Today we are starting concept 3, which is on the cell cycle and cancer. In this section, we're going to focus on the details or kind of the backstory of how it is as humans that we start out in this initial fetus structure, pretty human-like. We got all the parts and the pieces, and how do we get to this functioning human? How do we get to this little baby? And then throughout our lives, we continue developing into a, fully grown adult. So we're going to look at the process on the cellular level that gets us through all of these stages. I should really probably change the picture because I don't think any of you probably know who this is anymore. If you do know, come tell me who you think it is and I will give you a sucker. So that process going from our little fetus to a baby to a full-grown human all begins with a fertilized egg. So once our sperm cell fertilizes the egg, that fertilized egg we are going to call a zygote. So that new one cell structure is a zygote, and this is going to continue to go through cell division over and over again. So if you look at our picture over here, we have the one egg cell and the sperm cells, whichever one wins, they're going to fuse together to create a new one cell. We call that the zygote. So that new cell that we have, our zygote, is going to go through cell division through the process of mitosis that is going to give rise to many identical cells. So that one cell is just going to divide and divide and divide over again, creating a whole bunch of identical cells. So because we're creating identical cells in mitosis, we will need to go through the process of differentiation. And this is a process that creates special structures and functions depending on how these cells differentiate. And so in this, our specialized cells are going to become tissues, and then tissues become organs, organs become organ systems. And again, just a little more on the backstory. If we are looking at a fertilized egg, if you take anatomy, we will focus a whole lot on the structure of our developing egg and how it becomes fertilized and what the process is to create a fetus. So as we talk about the process of cell differentiation, we need to talk about the different cells that we have. We call these types of cells our stem cells, and these are undifferentiated cells that can become differentiated into one or more types of specialized cells. So I just want to pause and focus on why we're special, like why are we spending this time talking about stem cells opposed to just regular cells? Well, a normal cell, so if we're talking about a hair cell, when it divides, it can only become more hair cells, but these stem cells have the ability to become one or many other specialized types of cells. So if we look at our picture over here, we have these stem cells that are becoming liver cells. We have stem cells becoming heart cells or bone or brain cells. So it can become a whole bunch of different things, unlike all of the other cells. And we'll talk about this a little bit, but there's two different types of stem cells, and stem cell research can sometimes be controversial depending on which type we're talking about. But the first one is embryonic stem cells, and these are stem cells that have never differentiated and can become anything. And what is controversial here is if we think about this root word of embryonic, they're coming from embryos. So the only way to use embryonic stem cells is to get them from unborn babies. But on the positive side of embryonic stem cell research, we can make these cells into anything that we need. So if you have family members waiting, for a liver transplant, you don't have to wait for somebody else to die. We could just grow our own using these embryonic stem cells. The second type are adult stem cells, and these are cells that are found in adult bone marrow, and they are partially differentiated and only can become certain other things. So they're not going to be able to create anything in the body, but they can make more things than just our normal everyday cells. So if we look at cell differentiation, we also need to talk about cellular organization. So if we start the most complex things cells can form, we would call that an organism. And an organism is one individual member of a species. We move down from there, the next thing smaller than an organism is an organ system. And this is a set of organs that work together for a common function. So we could think of the digestive system. If we want to break the digestive system down smaller, we are going to talk about the individual organs within that. So we have our organ system here, which is a skeleton. And then if we break that down, we're looking at one single bone or one single organ. So an organ is a set of tissues working together for a common function. So we could think about the stomach for the digestive system, or a bone over here for our skeletal system. And then we have tissues as our group of cells working together for a common function. So we could have our muscle tissues that make up the stomach, or we could have osseous tissues that make up our bone. And then the smallest thing we have is our cell. This is our most basic unit of life that has all of the characteristics of life. So we've talked about that in the cell theory. So we have a muscle cell that makes up muscle tissue, tissue that makes up the organ, organs make up the organ systems, and a bunch of organ systems make up the organism. So as we talk about cells dividing, we're going to look at this at the whole process as a whole. And we call this the cell cycle. And this is a repeated pattern of growth, DNA duplication, and cell division that occurs in our eukaryotic cells. And if you remember, eukaryotes have a nucleus. So that includes things like us. And essentially, we can divide this down into two things, two main purposes of the cell cycle, and that is growth and repair. So I like to use the example of myself. I am done growing. I haven't gotten any taller since 8th grade. So even though I'm not growing as in like I'm not getting any taller, I'm still doing growth and repair because I lose skin cells every day. I lose hair cells every day. My liver is replacing itself. Those are normal things that our body goes through even if we're done growing. And the cell cycle can be divided up into three main phases. The first being interphase, which is cell growth. The second is cell division, which we call mitosis. And then lastly, cytokinesis, which is where our cytoplasm separates. So to start off the cell cycle, we are starting with interphase, and this is the growth phase of the cell cycle. And so this is where our cells are going to spend the majority of their time. But we can split interphase into three separate parts. The first is being G1, or the gap 1 phase. And during this, the cell is going to grow and make proteins. And that's what our G stands for. So gap, you can think of grows. The second part of interphase is the S phase, or the synthesis phase. And this is where DNA replication occurs, where we're going to double the number of chromosomes. So if we remember, DNA is the structure that makes you, and we call these big structures chromosomes. So we're going to get more of them. We're going to replicate during the S phase. And if we think about this word synthesis, synthesis means to make something. If you're synthesizing, you're making it. So we're making more DNA in the synthesis phase. And the last one is quite simple. We're just going to have another growth phase. So we have G2, or gap 2, which is where we're going to grow more and make more proteins in protein synthesis. So at the end of interphase, our cell is going to have 2 full sets of chromosomes. That way it's ready to be divided in half. So in order to understand what's going on in our cells as they're dividing, we need to talk about some DNA vocabulary, because DNA is a vital and important step of what happens throughout cell division. So our first structure that we're talking about is a chromosome. And this is kind of a general term. We can use it a lot, but it's talking about some sort of long continuous thread of DNA that consists of thousands of genes and regulatory information. So it's one big long strand that's containing thousands of genes that are going to make you, you. So we can define a gene as a section of DNA with instructions for making a protein. So we could have the gene for hair color. We could have the gene for eye color, the gene for height. Those are talking about one tiny little chunk of the DNA, whereas the chromosome is the whole piece of the DNA. Sorry, the notes get a little crazy here for some reason, but each human cell, all of our DNA is organized in the same manner, and we're organized into a total of 46 chromosomes. And those 46 chromosomes are organized into what we call homologous pairs. So we're going to see this prefix a lot, homologous, which means same. So homologous chromosomes. So there's actually 23 pairs for a total of 46. And if we read this little part over here, your chromosomes come in pairs because you're getting half from your mom and half from your dad. So if we look at a chromosome, again, chromosome is a general structure that talks about majority of our pieces of DNA. So we could call this entire thing here a chromosome. But if we try to be more specific, we can say that it is a chromatid. And this is 1/2 of a duplicated chromosome. So just one of these chunks of our X here, half of it, is a chromatid. Sister chromatids are the two of them together, and sisters are identical. Our next structure we call the centromere, and this is the region of condensed DNA that looks pinched. So it's where our little X crosses. The center piece here is the centromere. And our telomeres are the last piece. These are the ends of the DNA molecule. And I like to call them tails, so it kind of helps you remember this T word of telomeres. The tails are the ends of the DNA molecule. So they would technically be on both sides. We'd have telomeres at the top and the bottom of our structure. So in terms of what this looks like in our DNA, so if we look at this chunk we have over here, we have our dad version and our mom version. So we can call these our unduplicated homologous chromosomes. Yes, we have two, but one's from dad, one's from mom, so they are unduplicated. But we can still say they are homologous because we have these homos. We have the same set. We have the same gene, so dad's gene for hair color, mom's gene for hair color. So they're homologous but unduplicated. Next, we're going to go through the process of DNA replication, which happens during our S or synthesis phase, where we're going to make copies of our DNA that we started with, which means that we're going to end up with duplicated homologous chromosomes. So now we have two versions of dads and we have two versions of moms. And because these are copied versions, we are going to say that now that they're sister chromatids. They are identical copies, so they are sisters.